Hey guys, welcome back to Swaggy Science Studios.
I'm Mitch, and I'm the newest member of the Swaggy Science Studios.
We lost a few.
Taylor and Ellie had to leave, so they had to recruit me. Because I'm cool.
Facts
So,
about the
In
recruiting
this
part.
chapter, we're going to be learning about the discovery of DNA. So the major question is it a protein or a nucleic acid that contains the genetic material? So Frederick Griffith conducted the first major experiment that demonstrated that DNA as the genetic material. He used two strains of a particular type of pneumonia virus. He used a smooth and he used a rough. Once injected, the live S strain of the virus caused pneumonia in the mice. Okay? And then the live R strain did not cause pneumonia. And then what he did is he put a heated version of the S and it did not cause pneumonia. And then he put in a heated version of R and it did cause pneumonia. So we were all confused. So in the result, if the heated S strain is now harmless and the heated R is always, or in the live R is always harmless, and they were mixed together, the mice contracted pneumonia. Just wait. And then in conclusion of this, the transformation occurred that the live R strain was harmless and the live S strain was harmful. Okay. Avery or Oswald.
So Oswald Avery, he did an experiment to identify the molecules that did the transformation of the R strain into the S strain of the DNA. So basically he said that the R cells changed into S cells, like DNA replication, bringing the information of the disease causing the factor. So basically he said that the heated S strain turns into the live R strain because that becomes not harmful, and when you heat the R strain it becomes harmful, so they switch. And they change the shape from rough to smooth.
So, Alfred Hershey and Martha Chase. So everyone thought that protein was always genetic material, but they gave definite proof that DNA was genetic material and protein was not. They use bacteriophages to show where genetic information was stored. Those are viruses that infect bacteria. Hershey and Chase used radioactively tagged portions of the bacteriophage, which is phosphorus, which that is part of a DNA molecule, and sulfur, which is protein. So in conclusion, DNA, not protein, DNA provides genetic information to reproduce.
So the structure of DNA stands for deoxyribonucleic acid, and they're made out of nucleotides. So you have, the parts are 5 ribose sugars, a nitrogenous base, and a phosphorus group. So their 4 bases are adenine, thymine, cytosine, and guanine. So some vocabulary words are purines are a double ring of C and N atoms, which are A and G are purines. A and G are always going to be together. Or no, never going to.
Never
Gonna be together.
Because they are double bonded.
Yes, the double ring of
Hydrogen bonds.
right. And pyrimidines are a single ring of C and N atoms, which are C and T, and they're never going to be together. So, to decode the information of the DNA molecule cells, use RNA. And the job of RNA is to carry out the instructions from the DNA, what's decoded. So, whatever. Okay, so the RNA structure is single-stranded. It has 5 carbon ribose sugars, a phosphorus group, and a nitrogenous base. So basically it's the same, except it's single-stranded.
And also, the U, which is uracil, which is purine, is only an RNA, so that takes place of the thymine in DNA. So that's a big difference. So Edwin Chargaff So he was a guy that studied the composition of DNA in samples of different living organisms in 1950. So Chargaff's rule is in any sample of DNA, it's A to T and C to G. So the apple in the tree and the car in the garage.
That's how you can remember them. X-ray diffraction. So that's when equipment developed to take a picture of DNA's crystal structure. Many argue that this technique, this is not directly used by Watson and Crick, but it led to the actual discovery of DNA's structure. A vocab word, double helix. That is a shape of a DNA molecule, which is like people call it a twisted ladder / staircase.
So she kind of referenced Watson and Crick, which is our next topic. They were in the Nature magazine in 1953, a long time ago. They were credited for the discovering of the structure, so the double helix structure of DNA, and they published a short write-up of their work, so basically a little conclusion. And they said both deoxyribose sugar and phosphate groups make up the backbone of the DNA.
And the reason the write-up was short is there were a lot of things that went wrong during their experiments, so they didn't want to be known for doing wrong things and making all these mistakes. So they only talked about the good, which is pretty short.
Right.
Chickadee check.
So the nitrogenous bases make up the interior rungs of the ladder. So when you see the ladder thing, the hydrogen bonds hold the DNA strands together. They're the screws or the nails of the ladder. They're kind of the glue. They hold it all together, right?
And hydrogen bonds aren't that strong, so if we put it in
It's
real
like
life
Velcro.
perspective, it's Velcro. You can take it on, you put it off, blah, 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 blah.
But it's
it
good.
holds it together, and that's the main thing.
And a good thing is that it does kind of come off easy, because when we get into the DNA replication, they don't want it to be super hard to replicate.
break.
Right. So there are two hydrogen bonds shared between them.
So they fit like this, and the hydrogen bonds are right there,
Bing,
the-
bing.
in the middle.
The C and G partners with the three hydrogen bonds.
And it's like this. And it's not possible for A and C and T and G to be together because it's not chemically possible for this to happen because you can't have a cheese waggling in the wind.
Exactly.
No cheese wagglers. Okay. An example one would be, say, if you had a strand of DNA that was AACTG, you would have TTG, and so on, because I can't remember the pattern that I said.
Okay, so the orientation of the DNA. So the top strand is 5 carbons to 3 carbons. The bottom strand is 3 carbons to 5 carbons. So it like evens out. So each DNA strand is oppositely orientated. So like I said, it's 5, and then 3, and then 3 to 5. So it's opposite.
So the chromosome structure is next. So histones are proteins in the DNA. Nucleosomes are DNA and histones. A chromatin is several nucleosomes wounded together.
So now I'll be talking about DNA replication and the process of replication. So Watson and Crick did a hypothesis about the process of DNA replication. So in that hypothesis, they found semi-conservative replication. So one strand of new DNA with strands of parental DNA. So the replication steps we're now getting into. So in step number 1, it unwinds. The enzymes are involved, so the DNA helicase, it unwinds and unzips the DNA in the double helix.
Zoom like that.
Zoom right
Just
now.
cuts
So
it right
just
in
like
half.
So like if this was
Almost like a scissor.
together, it's cut
Zir-
right there. Okay?
Like a table saw.
Yeah.
And hydrogen
Writer's
bonds
saw.
begin to break apart, which causes the unzip. So like if you're unzipping your coat and it gets like that Y form, that's what it looks like. And the enzymes are involved in the RNA primase, and it adds an RNA primer, so the start point, on each DNA strand. So the second step is the base pairing. So enzymes are involved. So the DNA polymerase, it adds nucleotides to new DNA strands, so it like fits back together.
Construction.
Yes, the construction workers. So the leading strand replicates normally, and the lagging strand replicates backwards in the section so that it can match. And the Okazaki fragments are small segments of DNA made in pieces on the lagging strand only. So step 3 is the joining. So enzymes are involved in the DNA ligase. So it connects
Ligase.
ligase. Good job. So it connects the strands together. So it's like zipping your coat back up after you unzipped it. So you're too hot, splits.
So it goes
Too
like
cold,
this.
zip back up, and it becomes another strand.
Bing,
Exactly.
bing,
Bang,
bing.
and then zips back together.
About prokaryotic cell DNA replication versus eukaryotic cell DNA replication. And as we all know by now, prokaryotic cells have no nucleus, and eukaryotic cells do. And DNA replication happens in the nucleus, so it's kind of a big difference, right? So eukaryotic DNA replicates in multiple areas along the DNA strand, and prokaryotic DNA just replicates in one place. So that would be like if we were having road construction on a big road, we would have multiple workers going because that's a big long road,
In a
right?
eukaryotic
In eukaryotic
DNA.
DNA. And in prokaryotic, it'd just be like one worker's going along the road fixing the whole thing.
So now we will be talking about central dogma. So for the cells to read the information, it contains, or that is contained in the genes, so DNA, RNA, proteins. So the messenger RNA, which is the mRNA, so messenger is the M in it, and then RNA. The shape is uncoiled, it's single-stranded. The job of mRNA is to transfer information from the DNA to make proteins and travel from the nucleus to the ribosomes with the information. So it's like a messenger. The ribosomes RNA, so the rRNA, which is the R's for ribosomal, the shape is globular, so it's a big circle like a globe. And the job for that is the site of the protein assembly, so on the ribosome itself. Transfer RNA, so tRNA, transfer is the T, the shape is like a hairpin-like, and it's got 23 varieties. So the job, each variety of the tRNA bonds to a specific amino acid, and there are 23 varieties of amino acids. And it translates the information to be made into proteins. Wow. So now we're going to be talking about the transcription, which is the process of how RNA, is made from DNA. So the first step is the enzyme, the DNA helicase, and the DNA unzips in nucleus. Only the leading strand of the DNA used to make RNA. Then the lagging strand is not used to make the RNA. And in step number 2, the enzyme, the RNA polymerase, it adds the RNA base. Then the RNA processing is the next subject. So final, the mRNA does not code for every base on DNA. Not all information is necessary to make RNA.
So the introns is the intervening of sequences, which is not coded for.
So introns are cut out by special editing molecules. So it's like if I was editing this video, I'd cut out what I don't want. That's what they do.
Exons are remaining pieces of DNA which are coded for.
So exons hold actual information that need to be read. Okay. So our next topic is the genetic code.
Codon is the 3-base code in DNA or mRNA.
Yes.
Translation. Translation is the process of assembling proteins.
So tRNA molecules, these are interpreters of the mRNA sequence. tRNA helps translate language of amino acids to language of whole proteins. So step number 1, mRNA moves out of the nucleus and goes to the cytoplasm of the cell.
So anticondins are complementary to condon of mRNA found on tRNA.
So GAC is the codon for asparagic acids. CUG, which is CUG, is an anticodon. The second step, we were talking about the first step, and then we got the anticodon vocab. So the second step is the ribosome attaches to start code. So amino acids and the methionine, so the AUG on the mRNA to bond the amino acids together. Wow. This is just crazy to think about that this actually happens. The P site is where the anticodon is placed, and the A site is where the second anticodon is placed. Amino acids then bond, so the peptide bond joins the amino acids into proteins. The third step, when the ribosomes reach the stop code, the mRNA is released. The E site is where the amino acids exist in the ribosomes. So there's the different stop codes are UAA, UAG, and UGA. Go Georgia.
Okay, so I do this. Gene regulation and mutation. So the first part is prokaryote regulation. Gene regulation is controlling genes in response to environment. Operon is the operator, which contains genes for a metabolic pathway.
So T-operon controls the production of enzymes, and it makes the amino acids tryptophan, and the lac operons control the production of enzymes to digest sugar glycose.
So now we get into eukaryotic regulations. This is controlling the transcription, right? So genes, they need to be turned on and turned off sometimes, and they need to be done at the right time. It'd be like, when they're fixing things, you don't want something running and getting all messed up. So you have hoax genes. Hoax is short for homeobox, all right? Hoax genes are responsible for the general body patterns of most animals. So that's kind of responsible for the way that, most of us are made up of. And the mutation, we probably all know what mutation is, right? Well, it's a permanent change that occurs in the cell's DNA. Point mutation causes a chemical change in one base pair. So you have a couple types, 3 types. You have substitution, missense, and nonsense. Substitution is when one base is exchanged for another. Missense is when the wrong amino acid is coded for, and nonsense is when translation stops early. So that'd be like when they're fixing it, then they don't quite get it fixed. Not good. You have frame shift mutations, and so those are when the frame of the amino acid is changed due to extra or missing bases. There also is 3 types of frame shift mutations. So those are the insertion, deletion, and duplicate. So insertion is when you're adding an extra base. You can think of that as when you insert something, you're going to add. When you delete something, you're going to take it away. So that's the loss of a base. And when you duplicate, obviously you're going to add a duplicate base. So you're going to copy it. It's kind of simple there. So expanding mutations, these add repeating nucleotides to each generation. And there's only really one type of that, which is tandem repeat. So that's each generation is going to receive 2 or more repeated bases that every generation is added. Tandem means 2, so each generation is just going to receive 2. It's it's basically the same thing as duplicate, except we're getting 2 instead of 1 duplicate. So then we get to protein folding and stability. Genes must be read and decoded properly. If they're not, the protein is not going to be correct. You got to do things. If you're building a house, you got to build the foundation right, or the house is going to fall down. You got to build it right, or it's not going to function. So this is just the genes have to be read and decoded properly, or else the protein's not going to work the way you want it to. Okay, and if the protein does not fold correctly, like I said, it's going to malfunction. And so we're going to get into mutagens then. Mutagens, the environmental cause of mutations. So if you build your house the wrong way, and the wind comes in, well, the house is going to fall down. So that's the, kind of like the environmental cause. The mutations in the body cell, they are not passed on to the next generation, and mutations in the sperm and egg cell are passed on to the next generation. So mutations can be good or bad. That's how natural selection happens. And so yeah, that's
So this is
What we
it
got?
for chapter 12, molecular genetics. So we'll see you in chapter 13. Bye.